Okay, let's get started. So, hello everyone. My name is Xiao. I'm a second year student from Welch Lab. Thanks very much for showing up, and I hope you all are doing well at home. Today, I will be talking about my recent work. The topic is iterative refinement of cellular identity from single-cell data using online learning. Please feel free to ask questions. The talk is outlined as follows. There are four main sections: background, methods, results, and discussion. So let me first introduce you some background about single-cell RNA-seq technologies. So this figure demonstrates the key advantage of single-cell RNA-seq in comparison to the older bulk RNA-seq technologies. The bulk RNA-seq data displays an average of gene expression patterns across thousands to millions of cells, and this might obscure the biologically relevant differences between cells. In contrast, the single-cell RNA-seq enables gene expression to be studied at a single-cell level resolution, revealing the cellular heterogeneity. Next, let's look at how researchers determine the cell identity. So cells have long been qualitatively characterized by a combination of features such as morphology, cell surface proteins, and their broad function. So given multi-model single-cell data available, researchers are now able to quantitatively identify cell types and cell states using transcriptomic and epigenetic profiles. Here are three major challenges in integrating multi-model single-cell data. The first one is only one modality is available for each individual cell. Both similarities and differences among datasets are of interest, and the sample size is large and rapidly growing. Therefore, researchers in this area have put a lot of effort into addressing these issues, and a number of tools have been developed. So a paper published early this year listed top 3 batch correction methods after benchmarking 14 of them. They are Liger, which is our published work of our lab, Harmony, and Iterative version 3. Although they all show good performance, there are a few limitations. All of the 3 methods are not efficient in scaling to massive datasets and not able to incorporate new data without recalculating from scratch. Moreover, the Harmony is not designed for integrating multiple data modalities. So since our work is based on Liger, let's talk a bit more about it. The core of the Liger package is integrative non-negative matrix factorization, INF in short. As the figure shows, assuming we have n gene by cell input datasets, xi, of either same or different measurements of cells, they share the same set of genes, then our goal is to jointly decompose these matrices into shared matter gene factors W, dataset-specific matter gene factors Bi, and the cell factor loadings Hi. So the part on the left of the objective function focuses on the matrix reconstruction, while this part controls the sparsity. There are 2 parameters we need to specify, k and lambda. K is the number of dimensions of the latent space. In other words, how many metagenes we are looking for. Larger K leads to more factorized metagenes. K depends on the complexity of the biology underlying the data. So whereas the lambda determines how much we want to penalize the sparsity, so we use 5 by default. So before diving into the solution to IMF, let's first look at the existing algorithms designed for regular non-negative matrix factorization problem, so which is usually formulated as this. So we want to find the non-negative matrices W and H to properly reconstruct the matrix A. The problem itself is non-convex, but the subproblem where we solve for one component while keeping the others fixed is convex. Li and Song proposed multiplicative updating rules 2 decades ago. The idea was pioneering, but it offers no guaranteed convergence. Later on, to address this, researchers introduced 2 popular approaches in the block coordinate descent framework. The first one is ordinating non-negative least squares, ANLS, which updates the matrix blocks in an alternating fashion. The second one is hierarchical alternating least squares, which updates vector blocks. They are faster algorithms with guaranteed convergence. Multiplicative updating rules, ANLS and house, are all batch algorithms as they access a whole data set at iteration for training. So in Liger package, the batch algorithm ANLS was used for solving RNNF. So given n datasets, ni cells and m genes for dataset each dataset I, in order to minimize this object function, the matrix blocks hi, w, and vi are updated using this formula iteratively until convergence. So as the data size is rapidly growing, we're in need of a scalable tool for integrating single-cell data. Our work is inspired by the idea of online learning from my work paper, which is originally designed for regular NMF and sparse coding. As mentioned earlier, the iterative batch method accesses a whole dataset at each iteration, while the proposed online approach processes one data point or mini batch at a time. It provides fast convergence, it uses a fixed peak memory, and it does not have to recalculate from scratch in the presence of new data. So our goal here is to extend the online learning to the integrative non-negative matrix factorization, INMF. We envision using online IMF to integrate a single-cell dataset in 3 different scenarios. The figure here illustrates scenario 1. Assume the datasets are large and fully observed. The algorithm accesses mini batches from all datasets at the same time and repeatedly updates the meta-genes and cell-factor loadings. Each can be revisited throughout the multiple epochs of training. A key advantage of scenario 1 as compared to batch algorithm is that only a single mini batch needs to be in memory at a time. Depending on the available computational resources, it is controllable by manually specifying the size of the mini batch. Scenario 2 is demonstrated in the left-hand side. The input datasets arrive sequentially, and the online algorithm uses each cell exactly once to update the meta-genes without revisiting the data already seen. The key advantage of scenario 2 is that the factorization is efficiently refined as the new data arrives without requiring recalculation each time. On the right-hand side, we show scenario 3. It allows us to project new data into the latent space, more specifically the shared meta-gene W, already learned, without using the new data to update the meta-genes. In practice, we first use online IMF to learn the meta-genes as in scenario 1 and 2, then use a shared meta-genes W to calculate the cell factor loadings for new dataset. The third scenario is highly efficient in incorporating data. It allows users to query their data against a curated reference and provides increased robustness to dataset differences in newly arriving data. Now let's jump into the methods. In order to address optimization problem of INMF, we carry out these steps iteratively. At each iteration, we sample mini batches of cells from each dataset, solve for cell factor loadings using the meta genes WI and V, W and VI, then last step. Then we update the meta gene factors using the latest HI. So our strategy is to combine previously discussed ANLS and house methods. So for each sample mini batch, we update the cell factor loadings, the HI matrices using ANLS. So it is addressed by the block principal pivoting method developed by Kim and his colleagues. The formula is shown here. Then we update the managing factors using the house algorithm. So in this framework, there are 2n plus 1 times k vector blocks. We update each column vi and w using the displayed formula. Notably, as marked by the red boxes, the updates for w and vi depend on all of the previously seen data xi and their cell factor loadings hi. A naive implementation would require storing all the data as well as the cell factor loadings in memory. These are gene by cell and k by cell matrices, so hence it is not efficient at all to do so with a large number of cells. However, we notice that the house updates depend on the xi and hi only through the matrix products hi transpose, and xi hi transpose. In online IMF, let's assume we have mini batches from xi at iteration t. The size of each mini batch is pi, and the learned cell factor loadings are lowercase hi t. So we save hi transpose and xi hi transpose in matrices A and B. The dimension of A and B are relatively small. They are k by k and m by k elements respectively, allowing efficient storage and can be computed incrementally using this formula with incorporation of each new data point or mini batch xi at iteration t. Here is an overview of the online IMF algorithm we proposed. Given the inputs, we first specify k and lambda parameters and initialize the managings w and vi. Then for each iteration, we sample mini batches from each data set. We first obtain the cell factor loadings using ANLS, then updating matrices A and B. Use them to learn the managing factors w and vi through house. And in the end, we obtain the final HI using the latest managings through ANLS. So since we expect the inputs are too large to fit in the memory, the online IMF by design operates on the data saved on disk in HDF5 format. For example, when the mini byte size is 5,000 and 10,000, loading data from disk shown in blue adds minimal overheads as compared to loading directly from the memory as colored by yellow. So before I jump into the results, is there any questions about the background and methods? Okay, so let's move on. So I'm going to present the results in 4 parts. So in part 1, we compare the performance of online IMF and batch IMF. Then we benchmark online IMF along with other popular algorithms in part 2. So in the third part, we demonstrate online IMF on a large data set. Last but by no means the least, we show online IMF's ability to iteratively refine cell atlas. So let's start with part 1. For this part, we want to show online IMF converges faster and does not lose accuracy as compared to the batch IMF. we use the ANLS and the multiplicative method for comparison. The dataset we use are cells from adult mouse, frontal and posterior cortex. They are both single-cell RNA-seq datasets. The data collection is called Dropbase, created by Saunders and her colleagues. There are 9 different regions in total sequence. So for now, we are just using part of the data set. For online IMF, we tested different mini-byte sizes ranging from 1,000 to 200,000. We use k equals 40 and lambda equals 5. And we use 80% of the data for training and the rest of it for testing. So panel A and the panel B show that the online algorithm marked in red converges much more rapidly to a similar or better object function value compared to the batch methods, ANLS in green and the multiplicative in blue, on both training and the testing sets. In panel C, the box plot compares object function values achieved by online and batch algorithms after a fixed amount of training time. So we can see that the online algorithm leads to a significantly lower object function value within around 500 seconds. Then we run the algorithm using different choice of mini-byte size. From panel D and E, we see that the convergence behavior of the online IMF is nearly identical for mini-byte size from 1,000 to 10,000. And the panel F shows that for a fixed test set, the runtime needed to reach convergence remains relatively constant once the total number of cells exceeds approximately 50,000 cells. This behavior likely occurs because for a cell population of fixed complexity, for example, a tissue containing 10 cell types, only some fixed number of observations are required to effectively learn the meta genes. Therefore, using the entire dataset to update the meta genes at each iteration becomes increasingly inefficient as its size is larger than the minimum threshold needed to learn the meta gene. For the next experiment, we use the same cortex data plus human PBMCs and human pancreas data collections. The parameters for implementing online IMF are shown here. After we obtain the cell factor loadings, we visualize them using UMAP in the first and second coordinates. And we color the data points by datasets and by published cell type labels. So online IMF and batch IMF, ANLS here, produce very similar visualizations, suggesting that two approaches give very similar dataset alignment and cluster preservation. We will further confirm these qualitative observations using quantitative metrics next part. So in the second part, our goal here is to show online IMF yields single-cell data integration using less time and memory, but also producing comparable analysis results. So the methods used for benchmark here are batch IMF ANLS algorithm, Harmony, and the Surat version 3, the latest one. As for the benchmarking metrics, we use the time and peak memory usage for evaluating computational efficiency. To quantify the dataset alignment, we use the k-nearest neighbor batch effect test, the k-bet metric, and the alignment score. The k-bet uses a chi-square statistic to test the null hypothesis that the nearest neighbor distance in the aligned latent space do not differ according to batch. Thus, a higher average p-value indicates better data integration. The alignment score examines the mixing level in the local neighborhood of each cell. Proper batch correction produces a large alignment score near 1, while if there's no correction at all, then we will have score near 0. We use identical k-bet and alignment metric parameters to evaluate all approaches. To assess the clustering performance, we applied the Louvain community detection to the aligned latent spaces obtained by all methods. We then compare the results in clusters with the published cluster assignments using cluster purity and adjusted rand index, ARI. We utilize the same set of data for benchmarking the mouse cortex dataset, the 2 human PBMC datasets, and 8 human pancreas datasets. And the settings for running online IMF in string analysis is shown here. First of all, to benchmark time and peak memory usage, we generated 5 datasets of increasing sizes sampled from the mouse cortex data. So in panel A, we can see that the runtime required for online IMF, the red line, does not increase significantly as the size grows, and the peak memory usage is constant. Although not displayed here, we later found that the gap in runtime between the Harmony, green one, and our proposed method would become larger as the dataset size further increases, like over 1 million cells. The peak memory usage of online IMF with a minibyte size of 5,000 cells and 40 factors is around 350 megabytes, no matter how many cells in total are processed. The panel B and C shows the results on the human PBMCs and human pancreas, respectively. So as we can see here, the online and batch IMF algorithms align the PBMCs and pancreas datasets equally well and beating Harmony and Surat. So the online and batch IMF algorithm produce comparable clustering results to the other approaches in terms of cluster purity and ARI. So overall, the result of the analysis here indicates that the time and the memory efficiency of online IMF does not sacrifice the result quality. In part 3, we're going to showcase the application of online IMF on an even larger dataset using the fixed memory at peak. There are 9 single-cell RNA-seq datasets sequenced from 9 different mouse brain regions, totaling about 690,000 cells. So we ran the online algorithm for 3 epochs with minibyte size to be 5,000. The K and lambda we use here are 40 and 5 respectively. So it took about 24 minutes and around 350 megabytes of RAM on a laptop. I use MacBook Pro with i7 processors. So if we perform this analysis using our previously adopted batch IMF ANLS algorithm, it would have taken around 3 hours and required over 20 gigabytes of RAM. So we first visualize the embedded cells in panel A. The cells are colored by the published cell type labels, such as neuron, endothelial cells, microglia, oligodendrocytes, astrocytes, and etc. in the first and second UMAP coordinates, and separately displayed separately for all 9 mouse brain regions, such as cortex, cerebrum, hippocampus, striatum. So clearly, the online IMF algorithm retains the data structure, as we can see here. The cells within each class are grouped together very well. Then we use a factorization to group the cells into 40 different clusters by assigning each cell to the factor on which it has the largest loading, which we term the max factor loading assignment. So in panel B, we further examine differences in regional proportions of each cell cluster. The neurons and the oligodendrocytes show the most regional variation in composition. We notice that the total proportion of oligodendrocytes varies by region, but individual subgroups of oligodendrocytes are not region-specific. In contrast, the individual subgroups of neurons are highly regional specific, reflecting the diverse regional specializations in neuronal function. In panel C, we also investigated the biological properties of our cell factor loadings. The cluster assignments largely represents the cell type within the broad cell classes. As expected, the neuron shows most diversity with 8 subclusters. In contrast, macrophage, microglia, mitotic and neural cells each correspond to only one single cluster. So one of the most appealing properties of our online algorithm is the ability to incorporate new data points as they arrive. This capability is especially useful for large collaborative efforts to construct comprehensive cell atlas. So in part 4, we demonstrate online IMF by analyzing datasets that sequentially arrive. The cells used here came from the mouse primary motor cortex. They were generated by the Brain Initiative Cell Sensors Network, BICTN. So there are 8 datasets in total, and they come from 4 different modalities: the single-cell RNA-seq, single-nucleus RNA-seq, single-nucleus ataxic, and a single-nucleus methylation-seq. So there are about 400,000 cells in total. So as a setting here shows for running the online algorithm, it processes 5,000 cells per iteration, only one epoch for each dataset. and we still use k equals 40 and lambda equals 5. As shown in the figure, following scenario 2, our approach successfully incorporated each new single-cell or single-nucleus RNA-seq dataset, 6 in total, without revisiting previously processed cells. The cells are colored by the datasets. And these datasets are generated chronologically. We try to incorporate the single-cell nucleus ataxic dataset, which is a 7th dataset, in the same way, but unfortunately, it did not align as well as RNA datasets following scenario 2. This may be because the ataxic is a completely different modality, and thus scenario 2 may not be the best strategy for incorporating the ataxic data. Thus, we also integrate the single-nucleus ataxic data according to scenario 3 by the managing projection. So we first performed online IMF on the RNA-seq data, then used shared metagenes W to project the ataxic data into the same latent space as those RNA-seq data, as shown in panel B. And the data points from the ataxic data are colored in green. And then we can see here the new data points are well aligned to the previous data points colored in gray. Then we integrated the constellation data in the same way shown in panel C, and generating very satisfied results. So this strategy produced excellent integration results. In panel D, we were able to jointly identify 17 different cell types from all 8 datasets using marker genes reported in other studies. In summary, the online IMF has 3 key advantages. So first of all, it converges faster without loss in accuracy. Second, it decouples the memory usage from the whole dataset size. Therefore, it allows the researchers to carry out the single-cell data analysis integration on a personal laptop. Lastly, it is able to iteratively refine the cell identity as the single-cell datasets sequentially arrive. So there are still room for improvements. So specifically, we want to improve the performance when integrating a dissimilar type of datasets in scenario 2. So that pretty much covers my talk today, and I would like to say thanks to my mentor Josh and all the lab members for their continuous support throughout the project. And I will thank you all very much for your interest and attention. So here's a Q&As.
Yeah, there was a question from Damian Furman on how is online INMF implemented? Is it an R package or a Python?
Um, Currently, the online IMF algorithm is implemented in R package, and and to improve the computational speed, part of it is implemented in RCPP. And our lab member Lulu is currently working on making the R package into Python package. So that's a great question.
I think that's all the questions. And if anybody wanted, I'm sure you could take it offline. Thank you all for tuning in.
Yeah. Thanks very much. Have a good one.